Thanks everybody. For those of you who don't know me, my name is Joe Warren. I'm a faculty member in the School of Marine and Atmospheric Sciences at Stony Brook. I mostly do bioacoustical work, investigating where fish and zooplankton and some of their predators are in the ocean. But I also do a lot of net tows and sampling of the marine environment. So every now and then Mike Bottini sends me a picture of some random creature on the beach, and most of the time I don't know what it is, but sometimes I can figure it out. And so he'd asked me to give this talk for a couple of years, and I'm actually in New York at the time the meeting's held. So I'm going to take you on a tour of a variety of different organisms that I think many of you may not be aware of. If you are aware of them, great. I think they're amazing and fantastic. But there's no science in here. It's just pictures and videos that hopefully will work. So we're going to be looking at some of New York's oddest citizens. And normally I would throw up our departmental faculty photo for that, but we're going to focus on marine-only organisms. So how many people in the room have heard of salps? Okay, very good. How many people have heard of pyrosomes? Okay, a couple. Pteropods? All right, this is heteropods. All right, only a few. Great. Heteropods are my favorite animals, so we're going to spend about 35 minutes on them. So, and then I'm going to talk about some organisms that you probably won't find on the beaches of New York because they're in the mesopelagic, the deeper ocean. But if you were to tow a net deep enough, say in the Hudson Canyon or just off the shelf break, you would find some of these animals. So what is a salp? If you ask most people, they will describe it as this person from the Rockaways did, gelatinous blobs. So there's a variety of different species. These are organisms that are incredibly difficult to sample in the ocean because they're very patchy in their distribution, and they occur very irregularly in terms of abundance. You will go out and do a net tow and you'll get none of them, or you'll get 10,000 of them, and I'll show you some pictures of what that's like. They have a very unique sort of reproductive strategy life cycle that allows them to exploit, when conditions are good, their populations can really explode beyond exponential. It's actually geometric growth that they will experience. So salps, there are many different species. They all kind of have this similar structure. Let's see, is this the laser pointer? So they have a brain. Brain's a little tricky. Now, don't take this personally, but of all the organisms I'm going to show you here today, we are most closely related to this one. I know, it's kind of hurtful, right? Yeah. So they are urochordates. So of the zooplankton community, they are the ones that are phylogenetically most similar to us. And all they do is pump water through them, and then they filter out phytoplankton and other particles, store them in their gut, which is usually sort of bright red or orange, and they're transparent. And I can't remember if in any of these pictures, but you'll actually be able to see sometimes the reproductive cycle that these animals produce. So I always have to reference this because I can never remember which is a blastozoid and which is an oozoid. But salps typically have sort of two different forms that are either aggregate or solitary. And so aggregates often form these chains, and they tend to have like a sharp angle on either end. And so you'll see these chains out in the water column, and you could break them apart by hand, but they can be quite long. And then we have the solitary form, which looks more cylindrical. And so if you see one of those, you can get an idea just from the shape where they are. So what happens is if you're a blastozoid, which are the aggregate forms, each of you produces an oozoid. So you release one of these out, they grow, and depending on the species, this growth and sort of this reproductive cycle can take place in, kind of 5 to 25 days kind of thing. So they can regenerate, go through generations very quickly. When the conditions are good, this oozoid will actually have a chain of really small blastozoids within itself, and it can have anywhere from probably 30 to maybe even 100 of those organisms that then get released. They grow very quickly, and then each of those, spawns another set. So basically, each one of these can generate 50 to 100 offspring every week. So when conditions are good, these things will just go bonkers. And this is what happens when you do a net tow in a patch of salps. You get buckets and buckets and buckets of these. They're heavy. Sometimes we have to get a crane to lift the net out of the water because it's too heavy for us to pick up. We've had nets break from having so much biomass in their content. And it's, you know, there's other animals in there which we're excited about, but it's just this salp soup. And so it makes your processing time for these net hauls enormous. Like you just are doing buckets and buckets of these to try and find the non-salp creatures. Their body forms are actually, we think of them as kind of squishy, and they can be. This is one where it's lost most of its rigidity, but some of them can be quite rigid depending on what condition they're in. This is just another species that we've caught. This is not one that you'll find in New York. This is one from Australia. So there was a minke whale swimming around our Zodiac, and everybody was confused because I was reaching in to try and grab the giant salp, because at the point, this was the biggest salp I'd ever seen in my life. And then you get these guys. These are from the West Coast. They look like bunnies, right? They've got these little bunny ears. But these are maybe the size of my forearm, or can be up to the size of my forearm. And again, you know, these can dominate the biomass, and in terms of ecology, these organisms, because they boom and bust, can take a lot of carbon out of the surface ocean and drop it down into the sediments in a very short amount of time. So biogeochemists who are interested in how nutrients and carbon cycles through the ocean, we really have to account for these animals when we find them. Okay, so let's see if I can play a video. All the videos I'm going to show you are by me, except for this one, which is by Fish Guy. I know. But he's got the best footage of Long Island salps that I've seen. I've seen them when I'm diving, but I don't see this. So I think this is from 2 years ago. And so most of those animals at the top are individual salps. And then you can see the chains here in the surface ocean. The salp we have here in these waters is Salpa aspera. Its gut tends to be blue rather than red when you collect it. But we occasionally get this, you know, thick gelatinous soup here in New York. So, and you will see these, like that person in that first photo from the Rockaways. You know, they've found those animals on the beach, just walking on the beach at the rack line. All right, let's see, can I get out of the video? Yeah. So this won't play sound, but these are people. Salps are tasty, salps are great.
Salps are tasty, salps are great.
I don't know if just like us, they're Eurochordates.
Just like us, they're
All right, sorry. See if I can mute it. Is it gonna play? Yeah. So they are joining the Salp Sucker Society, which is the few, the brave, will eat a salp when we catch them on the boat. We rinse them off with seawater. It tastes like the world's worst Jell-O shot. It's just like, it's like a little slug of seawater, and it's chewy. You know, they're excited. See, John Hurwitz loves it. And we had multiple people on this trip join the society, so let me just skip past that. All right, so we're going to shift gears to a different gelatinous zooplankton. This is a juvenile stage of a pyrosome. So pyrosomes are sort of a colonial tunicate, which basically are a bunch of little cells that pump water, and they form these fairly rigid structures that look like this. We have a special name for them when we catch them on the boat. I'm not going to say it in public, but if you find me during the poster session, I'll tell you what we call them. And again, we can catch thousands, tens of thousands of these. And these animals have actually been shown to vertically migrate. Salps as well can move hundreds of meters vertically in the water column between night and day, which is kind of amazing given that they're, you know, fairly simple celled organisms. All right, these are just pictures of some random crustaceans that have evolved some various adaptations. We've got lobster larvae. We've got a shrimp that has moved its eye optical sensors onto its antenna, or not antenna, but stalked eye sockets. And then that's stomatopod, which is a really fun word to say, squat lobster larvae. And if you do a net tow out here in New York, this is sort of what you might find. So we have lots of copepods, we have some small krill, we have that darker crustacean is an amphipod. Then we have chaetognaths, which are sort of the long ribbon-like things. Those are arrowworms. Those, you can find chaetognaths that are maybe 15, 20 centimeters in length. But the good news is this is kind of a concentrated version because we've towed our net through the water column. So this has accumulated the animals over time. But how many people here have gone swimming in the ocean? Great, so you have all swallowed and eaten copepods. You've probably swallowed and eaten a bunch of cladocerans. Amphipods, maybe not. Chaetognaths, possibly. So these are, millimeter, 2 millimeter, 3 millimeter size organisms, but they drive the food web. So North Atlantic right whales feed exclusively on copepods, some of the smallest animals that we see. And they're really pretty amazing and beautiful. They come in all different sizes and shapes. This is one with egg sacs. Those blue dots are its eggs. We also, when we do our net tows, will catch juvenile animals of sort of fish you might recognize. So this is a filefish juvenile that we caught. Here are cladocerans. So cladocerans, you may be familiar with Daphnia, which is the freshwater water flea. That's a cladoceran. These are marine cladocerans. This is a tomopterid or a polychaete worm with really scary looking evil eyes. I think one of the reasons I love zooplankton is, when I was little, like most people, I wanted to go into space. I wanted to be an astronaut. And then you realize, like, you can see the most alien-looking things just right here off New York. All right, so this is a blurry picture. Apologies for it. On the very first slide, I had a picture of, anybody remember what was on the left of my title slide? Portuguese man-o'-war, right? Which is a type of siphonophore, which is another type of gelatinous zooplankton. This is a smaller siphonophore, maybe about this big in length, but still has this amazing blue coloration. In siphonophores, there's as natural historians, if you go to one of, Stephen Jay Gould, in one of his American naturalist essays, wrote a great article about siphonophores because we don't know if it's an organism or a colonial animal. And I think that debate is still in existence, whether it's many animals working together or if it's a single animal, because they have specialized cells that sting, they have specialized cells that pump water to make them move, they have digestive cells, and then they have the pneumatophore, which is this little air bubble or gas bubble that they use to control their position in the water column. Fun fact, a portion of my graduate work was funded because the Navy had torpedoes that were homing in on siphonophores rather than their targets because bubbles scatter sound very efficiently. So I have a soft spot for siphonophores. Leptocephali. So these are eel larvae, and these, again, can be incredibly long, 10 to 20 centimeters in length. Often you'll find them in sargassum beds. If you ever have sargassum wash up here during the summer, you can probably find some wrapped around there. This is a not quite in focus shot of a krill. So krill and copepods are kind of the key zooplankton that we have here in New York that feed most of the pelagic fish that are eating the lower food webs. And then we have occasional just amazingly beautiful small squid. So the nets that we use, we tend not to catch large animals, so we're going to be looking at the mesozoops. All right, this is perhaps the only zooplankton to feature in a major motion picture. So this is an amphipod, a hyperiid amphipod called Phronema, and it is in part the basis for the monster in the Alien movie franchises. You can see it's got some pretty crazy raptorial claws. It's got these huge translucent eyes. And these guys, like when we formalin them to preserve them, this is not preserved, this is just hanging out in a dish. They will fight and go bonkers. But H.R. Geiger, in part, based his drawings that the alien monster was based on these. So these animals are often found in these sort of houses, which they make from larvaceans, which is another type of gelatinous zooplankton that I'm not going to talk about. And occasionally you will find them inside their little house, and they just kind of hang out there. And what's really scary is when you find a house that's full of these, which are baby phronema. So we occasionally will find, a couple hundred larval or juvenile phronema in those houses, and we quickly preserve them so they don't take over the ship, and we have to fight them in the climax of the movie. Yeah, this is just to give you an idea of those claws. So these are, predatory animals that are kind of the meanest thing you will find in the ocean at the zooplankton level. All right, pteropods. So now we're going to shift gears and we're going to talk about pelagic snails. So these are snails or gastropods that spend their entire lives in the water column. They are not found on the seafloor. They are swimming about. And so we'll call them sea butterflies, sea angels. The shells come in a variety of different shapes, but they all have the feature where their foot has been modified to be a pair of wings that they can use to move about the ocean. And I think I've got a video here that'll kind of show you, Scooter McGillicuddy here. This is just in a petri dish on the boat, so it's trying, but they can move quite rapidly when they need to. So those shells are made of calcium carbonate, right? So these animals are particularly susceptible to things like ocean acidification, because that makes it harder for them to aggregate the carbon into their shell, or possibly causes their shells to dissolve more quickly. So some scientists are actually looking at shell thickness and shell mass as a way to kind of track how these animals are doing in the water column. Like I said, they come in all different sort of shapes, and again, here you can see the modified foot over on the right-hand side. Again, just the variety of shapes. And because the shells are very thin, they are fairly translucent. So this is just a larger pteropod trying to get out of the dish and failing. But let's get... the shells are beautiful. Most of the shells we get are broken because the net and the cod end, they bounce around a fair bit, but we can occasionally find good ones. This gives you an idea of sort of the range of sizes of shells you might see and species in a net. And this is just another example of how these animals how mobile they are. So the cool thing about the shells being translucent is you can actually see the organs of the animal inside. And I'll show you a video here and a series of photos where right now they're hiding in their shell, but they can emerge. They kind of look like bunny rabbits, right? Because they've got these, they're not ears, but it's part of their foot. These are their light sensory organs. And this is one that's fully out of its shell where it would be for feeding or locomotion. And they're just weird looking creatures. But I want you to remember this image in sort of this general shape where we've got eyes, we've got this nose, proboscis perhaps, we've got other parts of its foot, and this animal's just, coming out of its shell. I think here's a video where you can see, this is an animal. They actually have an operculum, so they have a hard shell that they close to keep themselves safe. And here's a video of it just coming out. And then I've got a better lit video here where I think you can see some of the organs pumping, right? So they're pretty amazing animals, and we're lucky that the shells are thin enough that we can see what's going on inside of them. And you can tell I like pteropods. I've got a lot of photos of them. Yeah. All right, so this is a gastropod that has gotten rid of its shell. So this is Cleone, sometimes called a sea angel. You can see them swimming around. They've got their foot. If you go on YouTube, I didn't download it, there is the crazy, when these guys eat, they basically have like a mess of tentacles that come out of this opening here and grab things. So they're sort of ambush predators to some extent. And this is my favorite zooplankton in the world. So this is a heteropod. I first saw this when I was a postdoc on a cruise in the Mediterranean, and I had no idea what I was looking at. And this is an animal that has evolved to basically decide its shell. I don't need the protection of my shell, so I'm just going to live outside my shell, but I'm going to keep the shell around, and it actually acts as a little bit of ballast for this animal while it swims around. So you remember that snail that I showed you? We've got a proboscis here. We've got eyes that actually have lenses. Well, I shouldn't call them eyes, but they're optical sensors with lenses, so they have some ability of vision. And then they've modified their foot to basically be a big oar or paddle, and then they keep this little bit of their shell around, which basically helps them orient in the water column. I've also described these as transparent Mr. Snuffleupagus from Sesame Street. I think that's a pretty good analogy. You can see the sort of the structure of the eyes here. Close-up of the proboscis. There's its shell. And then these come in a variety of different sizes, anywhere from a few inches to, yeah, again, 10, 15 centimeters. And they're found in, I've found them in every ocean I've been in. There are different species, so they have somewhat different shapes and styles. So this shell is a different shell than the other one. And then this one, I think this one looks kind of like Snoopy, but you know, maybe I'm anthropomorphizing. And again, this one is in sort of a sandwich-sized dish, so they're pretty. They can be pretty large, and again, that it uses that modified foot just as an oar, as somebody swimming in the ocean would. So. Okay, so how do we go out and catch these things? So what my group does, we use a variety of nets. Some of them are small. This is a midwater trawl, which is maybe 2 meters by 2 meters. This is a net that we can send down as deep as we have wire on the boat. So the deepest I think I've towed this is 1,800 meters, so more than a mile deep. We've also done projects with a net this size. This is called a mock nest. That's Tracy Sutton from Nova Southeastern University in Florida, inside for scale. This net is so large it barely fits out the A-frame of the boat. The reason it's a little more complicated is it's actually 6 nets on one frame, and we can control when these nets open and close with this system. So we can send it down to as deep as we want to go and open a net, bring it up 500 meters, close that net, so we know where those animals in that net were caught. Whereas with this net, it catches stuff on the way down, it catches stuff on the way up, so it's integrative, and we don't know where in the water column those animals were. If we want to go a mile deep in the ocean, we need about 6 hours of ship time. So we can only pay out, so to get 1,600 meters deep, we have to pay out about 3 kilometers of wire, and wire payout speeds are 20 meters per minute, 30 meters per minute. So just to put the wire out can take about an hour and a half. To haul the wire back in would take another hour and a half. And then we want to make sure we filter enough water so we get a significant number of animals, because in the deep, these animals are pretty sparse. How many people have seen pictures of like the deep scattering layer from acoustic systems, right? You see these, all right, I'm the only one who's seen those. But anyways, it looks like there's this carpet, right? We think of the deep scattering layer as this carpet of animals. But when you do the math, you might have 2 fish kind of in this stage area. Their densities are actually pretty low. And so we have to filter a lot of water, which just means like, so 6 hours of a boat this size, that's on the order of $15,000 of ship time just to do one tow, not counting people, not counting gear. So it's very laborious, and we come back from these trips with 6 tows, 10 tows. We don't have hundreds of tows. All right, so the twilight zone, what we're talking about is sort of the area that gets very little or no light in the ocean. Depending on who you talk to, it's anywhere from 200 meters down to 1,000 meters. This is midnight zone. Like whoever made this graph invented the midnight zone. I'd call most of this the twilight zone. And the animals that we find there have a lot of crazy adaptations to this environment, right? You think it's dark, we call it the midnight zone. There is light there because these animals have structures that tell us that there's light there. So these are myctophids, one of the most common fish that you will find in the ocean. When they come up on the nets, they're pretty beat up, so this animal has lost all its scales. But you can see they've got pretty big eyes relative to their body size, which doesn't make sense if it's completely dark. They also have a series of photophores along their body and along their ventral side. That's often how we identify species, is by counting photophores and the patterns of those photophores. And on many of those animals, those photophores luminesce, right? And the only reason you're going to have the only reason it makes sense to be a luminescent animal on your belly is so you're counter-shaded from visual predators that are looking up and looking for your shadow against light coming down. And so we know there are animals that use some form of vision in the deep ocean. All right, this is cyclothony. This is most likely, almost definitely, but not everybody agrees, the most abundant fish on our planet. It's called a bristlemouth. It's about yay big, and there are more of these fish than any other fish on our planet. There may be more of these than any other species in the ocean, although copepod people will argue about that. We've got, random deep sea fish with a little hitchhiker. So this is a parasitic copepod that has adapted to hang on by one little bit and suck out the life from that animal. Well, it won't suck out the life, but it's getting its nutrients from that animal. This is a hatchet fish. Again, really different body shape. Eyes represent a large portion of its body. Eyes are oriented so it can look up for things. These animals, it's hard to see here, they often have a translucent membrane right here, but they've evolved a very weird body shape, we think in part to avoid predation. And most of these animals have some form of teeth because they are ambush predators. So most of these fish, we think, are sort of just hanging out in the deep and waiting for a pressure signal or some form of signal for them to pounce. And then when they do, they've got some pretty good fangs, right? So these are viper fish. They have a very wide jaw and very sharp teeth. One of my first cruises, I caught a viper fish that had a myctophid in its mouth. And I was like so excited, and I was like, oh, we happened to catch this fish right in the middle of its feeding thing. How is that? That's crazy. And then somebody else on the boat was like, yeah, or it ate it in the net during the 4 hours that you were hauling it back up to the surface. And so probably the latter. Some of these animals have some interesting, so we've got luminescent photophores. We also have a luminescent eye patch right here, almost like an orca. And whether that's to lure in animals, we don't really know, but they glow in the dark. This is just another view of that luminescent patch. Again, eyes relative to the body, quite big. This is not an ambush predator, right? You look at these teeth here. This is another species of bristlemouth, agonostomatid. So this animal's feeding on some other way. All right, larval deep sea fish often have these little bags that they swim around in, and it's part of IDing them. So these are larval deep sea fish. This is a larval batfish, which has like a very flat ventral surface that still has its sort of larval sac with it. And here's why people came here. These are the most famous, the anglerfish. So anglerfish are incredibly rare in the deep ocean. I have caught 5, I think, in my career. And anglerfish have this lure on the top that often luminesces that they use to attract prey. With the exception of a few species, most anglerfish are about yay big, about 2 inches big. And those are the females, which are much, much larger than the males. The males are maybe a centimeter or so. For many of the species, when they reproduce, the male actually parasitizes onto the female. So it just clamps on to the body of the female, chews a hole, and then passes its sperm into the female, and then it withers away and dies. So whether that's an advanced reproductive strategy from humans, I'm not going to say, but you know, it's pretty crazy. This is a devilfish, another type of anglerfish. Its lure isn't quite as long, but it definitely luminesces. It's forked, and I'll show you another lure and explain why we think they're forked. This is one of the I think this might be the first anglerfish I caught, and so I took a lot of pictures of it. Craziness. Viperfish and dragonfish. So dragonfish have their lures on their chin. Again, they are often luminescent. And these animals can be a little bit longer. You get an idea of scale here. And then this is what one of the lures can look like for some of these animals. And what we think why it's evolved to this is so it looks like a small squid. And so we think those filaments are representing a tentacle, and that may be the same thing with that devilfish with the forked luminescent. I think that's it. Yeah, so you know, like everybody here understands, you know, you go out in nature and you can see amazing things. And so when you're walking the rack line, you never know what you're going to find. California, they actually found a football fish, which is an angler fish about yay big, and they found, I think, 3 of them washed up on the beaches there in the last 4 years. We don't know why those animals have come up and washed up. Nobody, to my knowledge, has found an anglerfish on Long Island, but it's certainly possible to do so. So thanks.
Got time for a few questions. Maybe you gave them all the answers.
That's fine.
When we were kids, we used to dig on the beach, sand crabs.
Yeah.
Sand crabs. And I don't see them anymore.
Like mole crabs?
They're about this big. They look like little.
So mole crabs are around, but I think like you've sent me some pictures where it's like I think they have a very boom or bust ecology, and I don't know if it's on a year. I know we've seen them because Mike sent me pictures of them, but yeah.
Joe, do you know what the jelly? What is the cause of severe jellyfish blooms? Some years there's no jellyfish in the bays, and then like at Long Beach, Noyak, you can't even go in the water.
So it's probably several different factors. So most of the things I showed you, we consider plankton, so they're drifters. So depending on the current velocities, they're going to get pushed or aggregated by physical oceanographic processes. They can control their position a little bit. They can move over short scales, but like over a tide cycle, they're gonna move with the currents. These animals can take advantage when conditions are good, but it has to be timed right with the critical mass of them to take advantage of those conditions, and also what other things might be consuming the prey in that area. So like I've done a bunch of work in Antarctica, and there salps and krill are in direct competition for the dominant phytoplankton, and krill can outcompete salps as long as they have a refuge for their juvenile stage. If they lose that winter sea ice that the juvenile krill can hide in, then the salps tend to do great. But the krill can kind of outcompete them if they're both there. So it's like we don't know the answer, but it's probably a variety of food availability, other predators for that food, and then physical oceanography. And the thing is, we don't have a lot of information on what things eat the various jellies. There's some turtle species will eat it. Now with eDNA techniques, they're starting to find a lot more gelatinous animals in stomachs. But I don't think we know whether some of that's like inadvertent consumption, right? Like in that pea soup from that first video, if you open your mouth, you're going to get some salps, whether you wanted to or not. But we think like seabirds, I've seen seabirds eating lion's mane jellyfish out on the ocean. So I think we don't really understand their role in the ecosystem very well.
Very luminous presentation, thank you. I have two questions. If we're like, you know, swimming in the ocean and we collect some of this water, can we see many of these like along the shore? That's number one. And number two, have you looked at any of your organisms as indicators of various conditions in the oceans? You know, like limnologists like Ruth Patrick have a whole retinue that you can look things up.
Yeah, so let me answer the second one first. So we have a couple different projects, one by New York State, one by another organization, where we're going out and monitoring the zooplankton assemblages in the sort of New York Bight waters. And one thing that's been shown, not from our data set because we don't have a long enough time period, but there's a NOAA data set going back 30 years where we know there are copepod species that prefer warm water versus cold water. And here in New York, we're seeing fewer cold water species and we're seeing more warm water species. So sort of that, you know, organism migration as an effect of climate change, you can do that with some of these lower trophic levels. Yeah, if you go to any body of water, you know, if you took a Nalgene and scooped it, you're probably going to get some copepods. If you want to do sort of a cheap and dirty net, just get pantyhose works pretty well in terms of the mesh size. Get a hoop or something to stretch it over. And you know, the more water you filter, the more animals you're going to have. But you could easily see copepods. Some of the copepods we get here are really hard to see with the naked eye, so you might need a microscope or a magnifier on your camera phone to see them. But like some of these videos, I shoot with an Olympus point and shoot that just has a really good macro mode. So.
Have you published anything on the the change in temperature?
I haven't. There are colleagues who have published some of those papers, and I can, if you email me, I can
Okay,
direct you.
Thanks.
Yep.
Okay, thanks, Joe.